Today we're going to talk about antigen presenting cells and how they're responsible for activating T cells. So antigen presenting cells can take many different forms, but the most common one that we know about and the most effective one we know about is the dendritic cell. And the dendritic cell is the only one we really know that can actually process that the antigen they find and take it and use it to activate T cells. So we have the dendritic cell, which was discovered by Ralph Steinman, and it's called a dendritic cell because when he saw these cells, he thought they looked a bit like what we know dendrites look like in the brain. So they're sort of spangly and have these projections coming out of the cell body. They're actually very different to dendrites, obviously, and they're very, very important. They're known as the sentinel cells of the immune system. They travel the body and look for foreign antigen so they can take it to T cells and to activate them. So what they actually do is, and we can see it here, is they have phagocytosis. They take part in phagocytosis. They have receptors on their cell surface, and what they do is they just sample cells. So they look at the MHC that's being expressed on the cell surface, and they process it. So, for example, this dendritic cell comes by and it has a look at the MHC that's being displayed by these epithelial cells, and this epithelial cell here has been invaded by a bacteria, and the bacteria has hijacked the replication mechanisms inside the cell and is using that to replicate itself. So the cell, as it's supposed to, is taking this protein that it's creating for the bacteria, and it's displaying it on the cell surface as MHC class 1, and which is really important because it's a great way for immune cells to tell not only if something is invaded, but if the cell's maybe over or under producing something, or is maybe a bit dangerous to us and needs to be killed. But in this case, the cell is producing cell components for this bacterial cell, and the dendritic cell comes on and sees that this MHC, this one in red, is being produced, and it says, well, actually through a toll-like receptor, it sees that this shouldn't be being made by this cell. This is foreign. So it recognizes this antigen as foreign. It takes the antigen from the receptor, because of course this isn't just this receptor here is more than just an antigen. What this actually is this is an MHC molecule, and inside in the MHC complex we've got this foreign antigen. So it's not actually, this whole thing isn't created by the cell. This is just takes a little sample of whatever it's producing, puts it with some MHC and displays it on the cell surface. And this dendritic cell comes along and it just samples some of this by phagocytosis, takes it in, and then it too displays its MHC on the cell surface with this MHC class 2, but at the same time it's able to recognize this as foreign. It sees a toll-like receptor and recognizes that this is not self, this is not healthy. So what it then does is it becomes activated, and it travels to the lymph node to find a T cell to tell all about it. These guys are not interested in fighting. These guys are all about raising the alarm, sort of like mast cells. They're not there to engage directly. They're just supposed to raise the alarm for other immune cells. So this dendritic cell travels all the way up to the lymph node, where it finds itself a T cell, and we refer to these T cells as naive T cells because they're not yet activated. They're not going to recognize self-antigen, but they're ready now with their T cell receptor, and they're equipped to. They're just waiting for it to be activated. So this T cell receptor here is highly specific to one certain antigen. So there will be hundreds of thousands of different kinds of T cells, naive T cells in the lymph node, and so the antigen presenting cell looks around for the T cell with the corresponding T cell receptor to the antigen it has. The dendritic cell displays this foreign antigen to the T cell, and I've labelled it as MHC foreign antigen peptide because it's not actually anything to do with the MHC. It's not, the T cell isn't interested in the MHC. It's seen MHC before, but what it's interested in is this foreign antigen. The dendritic cells process this antigen into a peptide, displayed it on its own MHC, and that's what the T cell is going to respond to. So the T cell receptor bonds to the MHC complex from the dendritic cell, and although that connection's made, that's not enough to stimulate activation of the T cell. What needs to happen in order for this T cell to become mature is it has to interact with other co-stimulatory molecules. So one of the things that exists on the surface of a T cell is CD4. So CD4 doesn't actually bind at the site of the MHC antigen recognition by the T cell receptor. It doesn't bind here, it binds on the MHC class 2 but not at the binding site. And what it does is it encourages this binding, it strengthens it. And CD3 is another one which is bound to the T cell receptor, and what it does is when it actually recognizes that the T cell receptor has bound the antigen, it starts sending excitatory signals into the cell, activating that cell, and that's what causes it to become a mature cell. And there's loads of these co-stimulatory molecules. I could talk about them all. CD4 actually is also involved in, it brings the IK enzyme in, which also aids the binding to the T cell receptor. But I've listed a couple here: CD80, CD26, interleukin 2 is really important, and CD40. These are all co-stimulatory molecules which take this MHC T cell receptor complex and turn it into an activated T cell. So it takes a lot more than just binding the foreign antigen. It has to have all these co-stimulatory molecules in check in order to actually have a naive T cell become an active T cell. So depending on whether you get CD4 or CD8, you can get CD4 cells and CD8 T cells. So the CD4 ones become things like T helper cells, and our CD8 cells become cytotoxic T cells, so killer T cells. But so in our example, we've used a CD4, so this now activated T cell by all these many different co-stimulatory molecules, it becomes active and starts to divide rapidly into lots and lots of different T cells. I think it's important to remember that the sort of overall function of this isn't just for the sake of making these T cells. You know, this cell's been invaded and we need to find the specific T cell that can interact with it. So there's lots and lots of T cells here with a huge range of T cell receptors, and this is the one that's been picked. So it'll go undergo rapid cell division in order to make more and more CD4 cells. So why make CD4 plus T cells? Well, these are T helper cells are the sort of common name for them. They're really important because they release cytokines which activate all sorts of things, which I'm sure you've probably heard of. Lots of macrophages, neutrophils and things like that are drawn to the site of infection and are encouraged to draw other things to the site of infection by cytokines. So they form part of the cytokine network and are so important. Depending on what co-stimulatory molecules act on the CD4 cell, it generally comes into different classifications. So you have T helper cells, but they can be T17, Th12, Th1. There's so many. They all have like wildly different functions, which you can read in. I can maybe go into further detail some other time. But if it then becomes successful and it interacts with antigen and has been a successful CD4 cell, and then becomes a T helper cell for whatever, like say it becomes a Th17 T cell, after a while, if it's still hanging around and hasn't died, it'll be acted on by interleukin 4 and will become a memory T cell. And so these memory T cells are really potent, so next time this kind of infection comes around, the response is much faster, because these memory cells are here to, you don't have to undergo this entire process again. As soon as they interact with that antigen, they're ready to go, and they've obviously been effective because they've survived the last round. So that's all about CD4 cells. Obviously I've said CD8 T cells are another common one made by this pathway, but Really important to remember though as well that this CD4 cell, although it can do all these things, is specific to this one antigen. So it has been activated by this antigen, and all the way from the start it's specific to this bacteria. So it's involved in this pathway, so it wouldn't necessarily be effective in any other kind of immune response. We have to remember that this immune response is highly specific. It's not like our macrophages or our neutrophils that are there to kill anything that moves. This is a specific response brought about by antigen presenting cells and the very specific T cell receptor. So while this might be a very effective T cell in fighting this one kind of bacteria, it wouldn't necessarily be able to fight a similar kind. But more on T cells later.